protein is actually kind of a nightmare to deal with.
It is a logistical nightmare. That is the perfect way to frame it. You see, if you think about fats and carbohydrates, they're relatively clean fuels.
Clean. What do you mean by that?
They're made of just three things: carbon, hydrogen, and oxygen. When your body burns them for energy, the waste products are carbon dioxide and water, CO2 and H2O.
Right. I breathe out the CO2, I pee out the water. Simple, clean burning.
Exactly. It's elegant. But protein has a fourth element, nitrogen. And nitrogen is the problem. It's like buying a beautiful new house that you love, but it comes with a literal unexploded bomb in the basement.
Okay, that's a strong analogy.
But it fits. You can use the house, the carbon skeleton of the amino acids, for all sorts of things, especially for energy. But first, you have to diffuse and safely remove that bomb. Because if you mess it up, if you just let that nitrogen go free, it immediately turns into ammonia. And ammonia is a potent neurotoxin.
So that is our mission for today's deep dive. We are going to act as the bomb disposal unit. We're going to trace the path of a piece of steak or maybe some tofu from the moment its amino acids enter your cells through the molecular shredding machines, onto these special transport trucks, and finally into the high-security chemical factory in the liver that neutralizes the threat once and for all.
We're going to break down the entire nitrogen disposal system. We'll look at how the body is constantly recycling itself, this amazing warrant system it uses to mark old proteins for death, and then we'll get to the urea cycle, which is, and I'm not exaggerating, one of the most beautiful and frankly expensive pieces of chemical engineering in the entire animal kingdom.
And we really need to keep emphasizing the stakes here. This isn't just, abstract chemistry on a blackboard. If these enzymes we're going to talk about have even a tiny defect, a single typo in the genetic code, you end up with these absolutely devastating conditions.
Yes. We're talking about newborns who appear perfectly healthy and then within days develop brain swelling, seizures, and suffer irreversible neurological damage.
That's terrifying.
It is. Disorders like Angelman syndrome, or a whole class of them called urea cycle defects. These pathways we're about to map out are often the only thing standing between a high-protein meal and, well, metabolic collapse.
Okay, let's start at the very beginning then. The why. Why do we even have this nitrogen problem in the first place? And it seems to start with this concept from the reading called nitrogen balance. When I first read that, it sounded like a bank account.
It is an accounting system. It's really the gold standard for measuring protein metabolism in a whole organism. The basic equation is super simple: nitrogen intake minus nitrogen output.
So my input is just what I eat, the protein I consume. The output is...
What?
Mostly urine.
Mostly urea in the urine, yes. But you also lose small amounts in your sweat, in your feces, even sloughed off skin cells. In a healthy, stable adult, someone like you or me, hopefully, we should be in what's called nitrogen equilibrium.
Input equals output.
Input equals output. We eat a certain amount of protein, our body breaks down about that same amount of its own protein, and we excrete that nitrogen. We stay the same weight, the same body composition. Everything is in balance.
But that balance can and does tip.
It absolutely does. Let's look at the first case, positive nitrogen balance. This is when your nitrogen intake is greater than your nitrogen output.
So I'm keeping the nitrogen, I'm holding on to it. Where is it going?
You're building.
Yeah.
You are actively synthesizing new tissue. Your body is taking that nitrogen and incorporating it into new proteins. We see this, obviously, in growing children. They are literally turning that dietary nitrogen into new bone matrix, new muscle, new organs.
Right, they have to.
We also see it in pregnant women, building a whole new person. And of course, the classic example is a bodybuilder or any athlete recovering from a heavy workout and building new muscle.
So when I see that guy at the gym chugging a massive whey protein shake after lifting weights, he's trying to intentionally force his body into a state of positive nitrogen balance.
That's the goal. He's providing the raw materials and the stimulus and attempting to convince his body to invest that nitrogen into new contractile proteins, yes. But then we have the dark side of this equation. Negative nitrogen balance.
Which is when output exceeds input.
Yeah.
We're losing more nitrogen than we're taking in.
And this is almost always a crisis state for the body. This is what happens during starvation. It happens in cases of advanced, untreated cancer or immediately following a major surgery or a severe burn.
What's the connection there?
The body senses a major threat or a profound lack of fuel. Now, unlike fat, which we store in adipose tissue, or carbohydrates, which we store as glycogen, we have no way to store amino acids. There is no protein tank.
So you can't just save it for later.
You can't. So the body makes a ruthless calculation. It starts raiding its own furniture to keep the fire going. It begins breaking down functional, essential proteins to get at the carbon skeletons for energy.
It eats its own muscle.
It eats its muscle, its organ tissue, its enzymes. It breaks down this functional tissue, uses the carbon for fuel, and it just dumps the nitrogen. If you see a patient in a hospital bed who is wasting away, that is negative nitrogen balance in real time. It's a catastrophic breakdown of the body's own structure.
That is a very grim image, but it sets the stage perfectly for this idea of protein turnover. Because I think I, and maybe a lot of our listeners, used to have this idea that my body was sort of like a statue. I built a bicep, and there it was. It just stayed there until I died.
Yeah.
But that's not true at all, is it?
Not even close. You are not a statue. You are a river. The overall shape might remain the same for a while, but the water molecules, the proteins, are constantly flowing through. The average human turns over about 1 to 2% of their total body protein every single day.
1 to 2% a day. That's a huge number. That means in, 50 to 100 days, I've theoretically replaced my entire body's worth of protein?
Mathematically, yes, you get a full replacement over that time. Now, in reality, some proteins stick around for a lot longer than others. We can categorize them based on their half-life.
Like radioactive decay, the time it takes for half of it to disappear.
Precisely. On one end of the spectrum, we have what we call housekeeping enzymes. These are the proteins that do the boring, steady, essential jobs, like the enzymes in glycolysis that are constantly burning sugar for baseline energy. They're built to last. They are very stable.
So they have long half-lives.
Very long, sometimes over 100 hours. The cell builds them, and they just sit there and do their job reliably day in and day out.
But then you have the other kind. The reading called them regulatory enzymes. But I started thinking of them as the divas.
The divas, or the control freaks. I like that. These are the proteins that carry critical short-term messages. They are the signals that tell a cell, grow now, or divide now, or stop metabolizing this fuel immediately.
You don't want those signals sticking around for too long.
You absolutely do not want those signals sticking around. You need to be able to turn them on, and then just as importantly, turn them off almost instantly. So these proteins are designed to be fragile. They have incredibly short half-lives, sometimes as short as 30 minutes or even less.
Wow. So the body spends a ton of energy to build this complex folded protein. It uses it for 30 minutes, and then it immediately destroys it.
It has to. It's a fundamental principle of control theory. Imagine if the growth signal got stuck in the on position.
You get uncontrolled growth. That's cancer.
Precisely. Rapid degradation is the price of metabolic control. And this brings us to the how. How did the cell actually destroy these proteins? We don't just, wait for them to fall apart from old age. We have two very distinct, very sophisticated demolition crews.
Okay, let's start with pathway number 1. The lysosome.
The lysosome. This is the ATP-independent pathway. I want you to picture a small stomach floating around inside your cells. It's basically a membrane bubble, a vesicle, and it's filled with acid and a cocktail of vicious protein-chewing enzymes called proteases.
And this is mostly for taking care of outside trash, right? Like stuff the cell swallows from the bloodstream.
Mostly, yes. It's great for degrading extracellular proteins, old cell surface receptors, and blood proteins. And there's a mechanism here for blood proteins that I find absolutely beautiful and answers the question, how does the liver know which proteins floating in the blood are old and need to be retired?
This was the expiration date concept in the source material. It has to do with a sugar molecule called sialic acid.
That's the one. Most proteins that circulate in your blood are actually glycoproteins, meaning they're decorated with complex sugar chains like little antennas. The very tip of that sugar chain, the last molecule, is sialic acid. Think of sialic acid like the rubber tread on a car tire.
Okay, I like that analogy. So when the protein is brand new, fresh from the factory, the tires are fresh, full tread.
Exactly. But every time that protein circulates through your body, every time it bumps into a vessel wall, or gets squeezed through a tiny capillary in your toe, or passes through the turbulent chambers of the heart, it experiences physical wear and tear. That friction literally knocks off some of the sialic acid residues.
The tread wears down over time.
The tread wears down, and sitting just underneath the sialic acid, the next sugar in the chain is a molecule called galactose. So as the sialic acid tread wears off, the underlying galactose becomes exposed.
And the liver is like the state trooper on the highway, checking everyone's tires.
That's a perfect way to put it.
Yeah.
Liver cells have specific receptors on their surface called Asial-like glycoprotein receptors. They're constantly scanning the blood. If they see a protein with exposed galactose, in other words, with bald tires, they know it's old and potentially unreliable.
And they just grab it.
They grab it, pull it into the cell via endocytosis, and throw it straight into the lysosome to be degraded and recycled.
That is unbelievably cool. It's a purely physical wear and tear sensor. It's not even a chemical timer. It's just mileage.
It's nature's way of ensuring quality control in the bloodstream. But that system is for, disposal. It's somewhat indiscriminate. For the delicate internal regulation we were talking about, for executing those short-lived diva enzymes, we need something much more precise. We need a sniper.
We need the sniper. The cytosolic machine, this is the big one. The ubiquitin proteasome pathway.
And this is the ATP-dependent pathway. So right away, we know it's going to cost energy. This doesn't happen in a bubbling acid pit. It happens right out in the open fluid of the cell, the cytosol. And it all relies on a tiny, amazing protein called ubiquitin.
Great name, ubiquitous, as in it's everywhere.
It is everywhere. It is found in yeast, in plants, in every animal tissue. It is one of the most highly conserved proteins known to science, which means evolution hit on this design billions of years ago and basically said, okay, this is perfect. Do not touch this. The ubiquitin in yeast and the ubiquitin in you are almost identical.
And its job is essentially to be the mark of death.
Yes. But it's not as simple as just walking up and slapping a sticker on a protein. This is a highly regulated, multi-step, legalistic process. It involves a team of 3 enzymes, E1, E2, and E3.
The source material called them the tagging team, which felt a bit too sporty for me. Reading about the mechanism, it felt much more bureaucratic.
It is absolutely bureaucratic. Let's use a legal analogy. Think of the first enzyme, E1, as the judge. E1 is the ubiquitin activating enzyme. Its job is to pick up a free-floating ubiquitin molecule and get it ready. But to do this, it requires energy. It has to burn a molecule of ATP.
So there's a filing fee just to start the paperwork.
There is a filing fee. You're spending energy just to get the warrant issued. So E1 activates the ubiquitin. Then it hands it off to the second enzyme, E2.
E2.
E2 is the ubiquitin conjugating enzyme. Let's call E2 the courier. It's the person who carries the signed death sentence. But E2 is blind. It has the weapon, the activated ubiquitin, but it has no idea who the target is.
That's where the third enzyme, E3, comes in.
E3 is the key. E3 is the ubiquitin ligase. Let's call E3 the detective. There are hundreds of different E3 ligases in our cells, and each one is a specialist. Each E3 is trained to recognize a very specific target protein that needs to be destroyed.
So it's looking for a misfolded protein or maybe a regulatory enzyme whose time is up.
Exactly. E3 is the matchmaker. It finds the target protein, grabs it, and at the same time it grabs the E2 courier who is holding the ubiquitin. E3 forces them together.
And it catalyzes the transfer of the ubiquitin from E2 onto the target protein.
Yes, specifically onto a lysine residue on the target. But here's the kicker. One ubiquitin isn't enough. One warrant might be a bureaucratic mistake. To actually send a protein to the execution chamber, you need a chain. This is called polyubiquitination.
So the process repeats.
The E1, E2, E3 cycle repeats itself, adding more ubiquitins onto the first one, forming a chain. And the signal for destruction is usually a chain of 4 or more ubiquitin molecules linked together in a specific way.
It's like a heavy chain literally dragging the prisoner down. So now we have our target protein. It's been identified, arrested, and now it has this long tail of ubiquitin attached to it. Where does it go?
It goes to the proteasome. And if you look at the structure of the proteasome, like in figure 28-3 from the text, it's a genuinely terrifying piece of molecular machinery. It looks like a mechanical trash compactor.
It's a cylinder, right, like a barrel.
It's a huge macromolecular cylinder made of 4 stacked rings. The top and bottom rings act as gates. They are locked. The proteasome won't let just anything wander inside because the inside of the central 2 rings is lined with shredding blades. These are protease active sites ready to chop up anything that enters.
So the polyubiquitin chain is the key to get through the gate.
That's exactly what it is. The polyubiquitin chain is recognized by a receptor on the gate of the proteasome. The gate recognizes the warrant, it unlocks and opens. An AT-pase motor unfolds the doomed protein. It has to be linearized, and then it's fed, thread-like, into the cylinder.
Like feeding a tree branch into a wood chipper.
A perfect analogy. And just before the protein goes all the way in a final act of efficiency, the ubiquitin tags are popped off and thrown back out into the cytosol to be reused for the next victim.
Efficiency. Even at the gallows.
Ruthless efficiency. The protein is then completely shredded into tiny peptides, about 7 to 9 amino acids long, and these are spit out the other side to be further broken down into individual amino acids. This entire mechanism is so fundamental to how a cell works that its discovery by Sikanover, Hershko, and Rose won the Nobel Prize in Chemistry back in 2004.
Okay, so we've done it. We've successfully shredded the protein. The carbon skeletons can be reused or burned for fuel. The bomb is now exposed. We have all these amino acids floating around, and our cells are stripping them down for energy. This process releases the nitrogen.
And now the clock starts ticking, because as soon as you release that nitrogen as an amine group, it wants to become ammonia, NH3, or in the pH of the cell, the ammonium ion, NH4 plus cell.
So let's get specific. Why is ammonia actually so bad for us? I mean, I know it smells terrible, like a dirty litter box, but what is it physically doing to my cells?
It's a profound metabolic poison, and it has a special, terrible affinity for the central nervous system. It's small, it's uncharged, and it crosses the blood-brain barrier effortlessly.
So it walks right into your brain.
It walks right in. Once it's in the brain, it causes chaos in a couple of ways. First, it messes with the delicate balance of neurotransmitters, specifically glutamate and glutamine. The brain tries to detoxify the ammonia by adding it to glutamate, making glutamine. But this depletes the brain's supply of glutamate, which is the most important excitatory neurotransmitter.
So you're losing your go signal.
You're losing your go signal. And at the same time, all this extra glutamine builds up inside the brain cells, the astrocytes. Glutamine is osmotically active, so it pulls water into the cells.
The brain cells swell up.
They swell. You get cerebral edema. On top of that, the reaction to detoxify ammonia also uses up alpha-ketoglutarate, a key intermediate in the Krebs cycle. So you're also disrupting the brain's primary energy-generating pathway.
Wow. So your brain literally runs out of power and swells up inside your skull.
Which leads directly to the symptoms we see in hyperammonemia. Lethargy, confusion, tremors, slurred speech, blurred vision, and eventually coma and death. So the body has a very, very strict rule. No free ammonia in the blood, ever.
But wait, we just said we are constantly breaking down protein in our muscles, especially after exercise. The muscles are far away from the liver, which is the only place that can really process this toxic waste. We have a major transport problem.
We do. We've generated a toxic byproduct in a peripheral tissue, and we need to get it to the central processing plant without poisoning the whole system along the way. We need a hazmat truck. We need a safe, stable container to move the nitrogen through the blood without it leaking toxic fumes.
And the body uses 2 main amino acids to do this.
2 primary carriers, yes. Glutamine and alanine.
Let's talk about glutamine first. The notes suggest that's the general purpose, all-purpose truck.
It is. In most tissues, like the brain, as we just discussed, or the lungs, or the gut, when free ammonia is produced, a critical enzyme called glutamine synthetase gets to work. It grabs the ammonia molecule. It then forces that ammonia onto a molecule of glutamate.
And that reaction turns glutamate into glutamine.
Exactly. Glutamine has 2 nitrogen groups instead of 1, but it's electrically neutral and, most importantly, non-toxic. It's like putting the radioactive waste into a thick, lead-lined box. The glutamine can then float peacefully and harmlessly through the blood until it reaches the liver or the kidneys to be safely unpacked.
But the muscles, they do something a bit different, a bit more clever. They use the glucose-alanine cycle. And honestly, this is one of those biochemical pathways that when you learn it, you just have to sit back and marvel at how interconnected everything is.
It is a truly beautiful example of inter-organ commerce and cooperation. Let's set the scene. Think about a muscle working really hard. Maybe you're out for a long run.
Okay, I'm running. My legs are burning. I'm burning a lot of sugar, a lot of glucose.
Right. Your muscles are breaking down glucose through glycolysis to get ATP for contraction. And the end product of that glycolysis is a molecule called pyruvate. So pyruvate starts to build up. But at the same time, because you're running hard for a long time, you're also entering a catabolic state. You're causing micro damage to the muscle fibers. You're breaking down some protein.
So I have 2 waste products piling up in my muscles. Pyruvate from sugar and nitrogen from protein.
The muscle has a brilliant solution that solves both problems at once. It takes that toxic nitrogen in the form of an amino group, and it uses an enzyme to glue it onto the pyruvate.
And pyruvate plus a nitrogen group equals
Alanine.
alanine. Alanine is just another harmless, non-toxic amino acid. It's safe to transport. So the newly made alanine hops out of the muscle cell and into the bloodstream where it rides all the way to the liver.
Okay, now the liver is the ultimate service center. It sees this alanine coming in from the blood. What does it do? The liver takes the alanine inside, First, it does the most important job. It strips the nitrogen off and sends that nitrogen directly to the disposal plant, the urea cycle, which we'll get to in a minute.
So the toxic waste has been delivered and handled. But what's left over when you take the nitrogen off of alanine?
You get pyruvate back, the original carbon skeleton.
The same molecule the muscle started with.
The very same. And what is one of the liver's superpowers? It can run a process called gluconeogenesis. It can take small molecules like pyruvate and, by spending its own energy, build them back up into a brand new molecule of glucose.
And then it just dumps that new glucose back into the blood.
It releases it back into the bloodstream to travel all the way back to the muscle to be used as fuel again.
That's incredible. It's a shuttle bus system. The muscle sends its trash, the nitrogen, to the liver, but it wraps the trash in a useful carbon skeleton. The liver unwraps it, throws away the trash, repaints the carbon skeleton, turns it back into valuable sugar, and sends it right back to the muscle to be burned for more energy.
It's a perfect division of labor. It allows the muscle to focus purely on generating force, while the liver, the master metabolic organ, handles the messy chemistry and foots the energy bill for the recycling program.
Okay, so the trucks have arrived at the liver. Glutamine and alanine are at the loading dock. Now we have to unload the cargo. This brings us to part 3, the chemistry of extraction. We need to get this nitrogen off the carriers and ready for disposal.
Right. We have nitrogen coming in on glutamine, on alanine, and from the breakdown of other amino acids within the liver itself. The first goal is to funnel all of this nitrogen from these many different sources into one single place. We use a process called transamination.
This was the ping-pong swap mechanism in the reading.
That's a great description of it. The enzymes that do this are called transaminases or sometimes aminotransferases. You might actually know them by their clinical names, ALT and AST.
Oh, the liver enzymes. Those are the ones my doctor checks on a blood test to see if my liver's damaged.
Exactly. Alanine transaminase, ALT, and aspartate transaminase, AST. When liver cells die, they burst and spill these enzymes into the blood, which is why they are such sensitive markers for liver damage. But normally they are inside the cell working hard, and their job is to move nitrogen. Specifically, to move an alpha-eno group from any amino acid onto a single universal collector molecule, alpha-ketoglutarate.
So alpha-ketoglutarate is the bucket we're collecting all the nitrogen in.
It's the designated bucket. And when you throw a nitrogen group into that alpha-ketoglutarate bucket, it becomes L-glutamate. So alanine gives its nitrogen to alpha-ketoglutarate, creating glutamate. Aspartate gives its nitrogen, creating glutamate. It's all channeled into glutamate.
Why is it called a ping-pong mechanism?
It's because of the way the enzyme works. It doesn't bind both the amino acid and the alpha-ketoglutarate at the same time. It's a two-step process. First, it grabs the incoming amino acid, say alanine, ping. It steals the nitrogen from it and releases the leftover carbon skeleton, which is pyruvate.
Okay, so now the enzyme itself is holding the nitrogen. It's like a hot potato.
It is temporarily stuck to the enzyme, and it's actually bound to a critical cofactor, a helper molecule called pyridoxal phosphate, or PLP.
Which our bodies make from vitamin B6.
Correct. This is a great example of why vitamins are so important. Without sufficient vitamin B6, you cannot make PLP, and without PLP, you cannot handle protein. Your entire nitrogen metabolism would just stop.
So how does the PLP hold the nitrogen? The text mentions something called a Schiff base. That sounds like a terrifying flashback to organic chemistry class. What does that actually mean in simple terms?
Don't panic. A Schiff base is just a special but reversible type of double bond between a nitrogen atom and a carbon atom. Think of it as a temporary anchor. The PLP forms this Schiff base with the incoming amino group, anchoring it firmly in the enzyme's active site.
Okay, so the enzyme uses the vitamin B6 anchor to hold the nitrogen. Then what?
Then the first part of the molecule is released, and the second molecule, our bucket, alpha-ketoglutarate, comes in, PONG. The enzyme then transfers the nitrogen from the PLP anchor over to the alpha-ketoglutarate. The anchor releases, and the new product, L-glutamate, floats away.
So to summarize, we've taken nitrogen from all over the place, from alanine, from aspartate, from leucine, you name it, and we've used this ping-pong mechanism to funnel it all into one molecule, glutamate. It's like a metabolic consolidation loan.
That's a perfect description. But now we have a new problem. The nitrogen is consolidated, but it's still attached to something. To get into the urea cycle, we need it to be free. We need to rip it off of glutamate entirely.
And this next step is called oxidative deamination.
Correct. The star enzyme here is L-glutamate dehydrogenase, or GDH. This enzyme is a beast. And it's located in a very specific place, inside the mitochondria of the liver cells. It takes that L-glutamate we just made, and through an oxidation reaction, it literally strips the amine group off.
The result of that is free ammonia.
Free ammonium ion, NH4 plus AM, right there inside the mitochondrion. And this is the danger moment. This is the whole point of the process. We have finally released the toxic gas, but we've done it inside a contained, high-security vault. We must process it immediately, right where it stands.
This brings us to the main event, the centerpiece of our deep dive, the urea cycle, the waste processing plant itself.
And before we walk through the 5 steps, I just want to pause for a second on the evolutionary context here, because not every animal on Earth goes to this much trouble.
Right, the reading mentioned that fish, for example, they don't have a urea cycle, do they?
Most don't, no. Fish are what we call ammonotelic. They produce ammonia just like we do, but they live in an ocean. They live in a massive body of water, so they just excrete the ammonia directly from their gills right into the surrounding water. It's instantly diluted to a harmless concentration.
Ah, the solution to pollution is dilution.
For them, it works perfectly. But now, think about a bird or a reptile. They lay eggs, hard-shelled eggs. If the embryo inside that sealed container produced toxic, water-soluble ammonia, it would quickly poison itself and die. It can't pee it out because there's nowhere for the pee to go inside the egg.
So they need a solid waste product.
They need a solid, non-toxic waste product. Birds are uricotelic. They spend even more energy than we do to turn their nitrogen waste into uric acid, which is that white, chalky paste you see in bird droppings. It's insoluble in water, so it just precipitates out as a harmless solid inside the egg.
As a bonus, it's light, which is great for flying later on.
Exactly. But we are mammals. We develop inside a womb with a placenta, and we drink water. We are ureotelic. We needed a compromise. We needed a molecule that was highly water-soluble so we could excrete it in urine, but it also had to be non-toxic so it wouldn't kill us while it was being concentrated in our bladder. And that magic molecule is urea.
But magic, as you said, comes with a pretty big price tag.
A very hefty one. The urea cycle is an energy-demanding process, but it's the price of admission for being a terrestrial mammal.
Okay, let's walk the factory floor. The whole process takes place in the liver, and it spans 2 different cellular compartments. 5 steps. Step 1 happens deep inside in the mitochondria.
This is the committed, rate-limiting step of the whole cycle. The enzyme is called carbamoyl phosphate synthetase erythrolyse, or CPSI.
CPSI. Sounds like a crime scene investigation unit.
It's kind of fitting because its job is to contain a dangerous substance. It takes a molecule of CO2, a molecule of that free ammonia we just generated, and it fuses them together. But to do this, to force this reaction to happen, it has to burn 2 molecules of ATP.
Wait, 2 ATP just to start the first step. Usually enzymes use 1 ATP, if any.
This is an incredibly energy-expensive reaction. The product is a high-energy molecule called carbamoyl phosphate. The body is essentially paying double the market rate because it is absolutely desperate to trap that free ammonia and get it into a more stable form.
And there's a critical safety lock on this enzyme, right? It doesn't just run all the time, wasting energy.
That's right. If there's no protein being broken down, you don't want the factory running. So CPSI requires the presence of an allosteric activator molecule called N-acetylglutamate.
Okay, explain allosteric activator like I'm 5.
Think of the CPSI enzyme as a high-security vault door. You have the electricity to run the motor, that's the ATP, but the door will not open unless the bank manager is physically present to scan his thumbprint on a separate pad.
And the bank manager is N-acetylglutamate.
Yes. And the manager, N-acetylglutamate, only gets made and shows up to work when glutamate levels in the liver are high, which only happens when you've just eaten a steak and a flood of amino acids is arriving. No steak, no high glutamate, no manager. The vault door stays closed. It's a feed-forward activation system.
Very smart. Okay. Step 2. We're still in the mitochondria. We have our high-energy carbamoyl phosphate.
Now we combine it with a carrier molecule called ornithine, using the enzyme ornithine transcarbamylase, or OTC.
Ornithine is one of those weird amino acids, right? It's not one of the 20 used to build proteins from our DNA code.
Correct. It's a dedicated worker for this cycle, not a building block. Its job is to recycle. So ornithine plus the carbamoyl phosphate group gives us a new, larger molecule called citrulline.
Citrulline. I think I've seen that in pre-workout supplements.
You have. It was first isolated from watermelon juice, Citrullus vulgaris. Now, here is the big twist in the plot. The citrulline that was just made is now transported out of the mitochondria.
It leaves the inner sanctum and goes out into the main cell fluid, the cytosol. Why swap locations in the middle of the process?
It's most likely a containment and resource management strategy. You keep the most dangerous step, the handling of free ammonia in the deep vault of the mitochondria. Then you move the less dangerous intermediates out into the cytosol where other necessary components, like our next nitrogen donor, are readily available.
Okay, so we're at step 3. We are now in the cytosol. We have citrulline. We need to add a second nitrogen to build our final urea molecule. We got the first one from free ammonia. Where does the second one come from?
It comes from another amino acid, aspartate. The enzyme arginocessanate synthetase grabs the citrulline, grabs an aspartate, and fuses them together.
And let me guess. This step also costs energy.
It does. It costs another molecule of ATP, but this time, to really drive the reaction forward, it burns the ATP all the way down to AMP and pyrophosphate. That pyrophosphate is then immediately broken apart, which releases even more energy.
So energetically, breaking ATP to AMP is like burning 2 ATPs to ADP.
Correct. It's the equivalent of spending 2 more high-energy phosphate bonds. So if you're keeping score at home, we are now 4 high-energy bonds in the hole. We are spending an absolute fortune to build this waste product. The new combined molecule is called arginocessanate.
On to step 4. The enzyme is arginocessanate lyase. A lyase is an enzyme that cuts things.
Exactly. It cleaves the arginocessanate. It's a clever cut. It keeps the part that has all the nitrogen forming the amino acid arginine, and it throws away the carbon skeleton of the aspartate that we used. That carbon skeleton is a molecule called fumarate.
Wait a minute, fumarate. I definitely know that name. That's an intermediate in the Krebs cycle, the central energy-producing cycle in the cell.
It is. This is a beautiful point of integration, sometimes called the Krebs bicycle. The urea cycle doesn't just happen in isolation. It throws this fumarate molecule over the fence to the Krebs cycle, which can then use it to generate some energy back. It's like getting a small rebate on the massive energy bill we just paid.
Very cool.
Yeah.
Okay, step 5, the final step, the payoff. The enzyme is arginase.
Arginase is only found in the liver, which is why only the liver can make urea. This enzyme takes the arginine we just made, adds a water molecule to it, and it snips off the end of the molecule. That little piece it snips off is urea.
Success. We've made our non-toxic, water-soluble nitrogen waste product.
We have. The urea molecule, with its 2 nitrogen atoms, now floats away, diffuses into the bloodstream, travels to the kidneys, and is excreted in the urine. And the piece of the arginine molecule that's left behind, it's ornithine.
The exact same molecule we started with back in step 2.
The very same, which is then transported back into the mitochondria to pick up the next load of carbamoyl phosphate.
The circle is complete. The carrier has been regenerated.
And the factory is ready for the next shipment of nitrogen.
It truly is an elegant process. But hearing you break down the energy cost, 4 high-energy phosphate bonds for every single molecule of urea, it really drives home how serious the body is about this. It prioritizes safety far above metabolic efficiency.
Always, when it comes to ammonia, there is no room for error.
But what happens when the factory does break? We talked about the stakes at the beginning. Let's get into the hard part. Part 5, the metabolic disorders.
This is the clinical reality, and it can be incredibly tough. A urea cycle disorder, or UCD, means that one of these 5 enzymes, or one of the transporters, has a genetic defect. It doesn't work properly. Maybe it works at 50% capacity, or 10%, or not at all.
And no matter which step is broken, the result is always the same. Hyperammonemia.
High blood ammonia. The factory line backs up, and the raw, toxic material ammonia spills out into the rest of the body. Imagine a newborn baby. For the first 24, maybe 48 hours, they seem fine. They're just drinking colostrum. But then the mother's milk comes in, which is higher in protein. They start refusing to feed. They get very sleepy, lethargic. They start vomiting.
Which, as a new parent, must sound like almost anything. A common cold, the flu, sepsis.
Exactly. It is tragically and frequently misdiagnosed at first. But inside that baby, the ammonia level is spiking to 10, 20, even 100 times the normal level. It's crossing into the brain, causing the swelling we talked about. If you don't recognize it and intervene immediately, the child can slip into a coma and suffer permanent brain damage.
Let's talk about a specific one you mentioned, hyperammonemia type 2, the OTC defect.
This is the most common of UCDs. It's a defect in step 2 of the cycle, the enzyme ornithine transcarbamylase, which works inside the mitochondria. And what's clinically important is that the gene for OTC is on the X chromosome.
It's X-linked, which means it mostly affects boys much more severely.
Boys get the severe neonatal form because they only have one X chromosome. If that copy of the gene is broken, they have 0 functional enzyme. They are the babies who present in the first week of life with catastrophic ammonia levels that can be fatal.
But girls, or women, have two X chromosomes, so they can be carriers.
And this is where it gets fascinating and also terrifying. A woman might have one good gene and one bad gene. Because of random X inactivation, she might produce, say, 50% of the normal amount of OTC enzyme. For most of her life, living on a normal diet, that's enough. She's completely asymptomatic.
But then, something happens.
Then something happens that pushes the system. Maybe she decides to go on a very high-protein, low-carb diet. Or she has major surgery. Or she just has a massive Thanksgiving turkey dinner.
She creates a huge nitrogen load that her 50% capacity factory just cannot handle.
The system gets overwhelmed, the ammonia starts to build up, and suddenly this otherwise healthy woman becomes confused, agitated, she's slurring her speech. She might present to the emergency room and be misdiagnosed with a stroke or drug overdose or a psychiatric episode, when in reality, she's being poisoned by her own dinner.
That is just wild. And there was another one in the notes with a really strange and specific physical symptom, arginocystinic aciduria.
Right, that's a defect in step 4, the lyase enzyme. Besides all the severe neurological and ammonia issues, these patients often have a condition called trichorexis nodosa.
That sounds like a spell from Harry Potter.
It laughs.
It does. But it actually describes their hair. For reasons we don't fully understand, the buildup of arginosuccinate and the relative lack of arginine makes the hair shaft weak and brittle. It looks like little white tufts or nodes along the hair. It's dry, it breaks off easily, often close to the scalp. So if a physician sees a lethargic, vomiting child who also has this unusual tufted, brittle hair, a bell should go off in their head immediately. Think urea cycle.
So given how fast the brain damage can happen, how do we catch this before it's too late?
Modern technology has been a lifesaver here. The answer is tandem mass spectrometry.
Mass spec.
It's now part of the standard newborn screening panel in most developed countries. You take one drop of blood from the baby's heel on a piece of filter paper. The mass spec machine can, in just a couple of minutes, precisely weigh all the different amino acids and other metabolites in that blood spot. It can see, for example, oh, wow, the level of citrulline in this baby's blood is 1,000 times normal.
Which means the blockage in the factory line must be right after citrulline is made. Exactly. It points directly to a defect in the arginosuccinate synthetase enzyme, step 3. It can pinpoint exactly which enzyme is broken based on which intermediate molecule is piling up behind the dam. We can now diagnose these disorders before the baby even shows the first symptom.
And if you do catch it that early, what's the treatment?
The cornerstone of management is diet, a very strict, low-protein diet for life, and frequent small meals to make sure the person never enters that catabolic negative nitrogen balance state where they start breaking down their own muscle. We also have drugs, so-called ammonia scavengers.
Oh, they do.
Molecules like sodium phenylbutyrate act as chemical sponges. They provide an alternative pathway for nitrogen to be captured and excreted from the body, completely bypassing the broken urea cycle. It's a chemical workaround.
And the future? Is it all just diet and drugs, or is there hope for a cure, like gene therapy?
Gene therapy is the holy grail for these disorders. The liver is actually a really good target for it because it's a single organ and it naturally filters the blood. The idea is to take a harmless virus, an adenoviral vector, and put the correct functional DNA for the OTC enzyme inside it. Then you inject this virus into the patient.
And the virus infects the liver cells and delivers the correct gene.
It delivers the correct blueprint, and hopefully the liver cells start producing the missing enzyme. You restart the factory. We have successfully done this in animal models. There are human clinical trials that have shown some promise, but also some significant challenges. It is not a routine cure yet, but it is where the science is heading.
It's just amazing to think about that whole journey. We started with a burger, we went through a molecular wood chipper, we rode a shuttle bus system between organs, we toured a high-energy, high-security factory, and we ended up discussing CRISPR and gene editing.
It really is the full spectrum of biology, from nutrition all the way to cutting-edge medicine.
So as we wrap up here, I find I'm still stuck on that energy cost. 4 high-energy phosphate bonds per molecule of urea.
It still bothers you, the inefficiency.
It does. It just seems so wasteful. Nature is usually so stingy with ATP. It counts every single molecule, and here it is just throwing it away to make garbage.
Let's reframe the question. Think of it this way. What is the cost of not doing it? The cost is a non-functional, swollen, dead brain. Evolution works on a good enough to survive and reproduce principle, but when it comes to protecting the central nervous system, it doesn't cut corners. It over-engineers the safety systems.
So we pay a constant metabolic tax to keep the lights on upstairs.
That's it, exactly. It is a profound reminder that biological efficiency isn't always about saving calories. Sometimes, the most efficient strategy is the one that guarantees robust, long-term survival, even if it's expensive in the short term.
Well, on that note, I think I'm going to go eat some lunch.
Yeah.
But I might chew a little slower this time and have a little more respect and gratitude for what my liver is about to do for me.
Yeah. Respect the liver. It's probably the hardest working, least appreciated organ you have.
Thanks for listening to The Deep Dive. We'll see you next time.